All right, great. Well, thanks, Connie. I'll try and run through these slides fairly quickly. As Scott said, we've been around for a while. You haven't seen the end of us yet, I'm afraid. So a little update on behalf of Heng Hong and I, representing the TGx-DDI transcriptomic biomarker team. This team gets a prize for the most dedicated people ever because they have been doing this for a very long time. Not only that, but we've welcomed new members on board over the last years. So, we're all really committed to taking this to the end. So I want to thank every person on this list, and especially Cyril and Chrissy and Connie for helping to keep this ship moving forward. So why the TGx-DDI biomarker? TGx stands for toxicogenomics and DDI stands for DNA damage inducing. Well, we know that genotoxicity is a key driver of cancer. And so we have these standard tests for chromosome damage and mutation that you're all aware of, the Ames assay, in vitro mammalian gene tox assays. And then if one of these are positive, we go in vivo. But these methods in vitro have serious limitations. They tend to be highly sensitive, but they lack specificity. This means that they have high rates of misleading positives. A misleading positive is one that is positive in vitro, but doesn't cause cancer or genotoxicity in vivo. And they also lack efficiency. The Ames assay, for example, can't be integrated with other tests, and they don't provide information on the mechanisms causing genotoxicity. And so many, many years ago, the committee decided that there's a need for higher throughput genotoxicity tests that provide mechanistic information in human relevant cell culture models. So this slide is that kind of outline for the rest of the talk. This is the framework that's typically used for moving a transcriptomic biomarker or any biomarker forward towards regulatory acceptance. So there's the biomarker discovery phase, there's establishing the context of use, and then we go down these two sort of prongs towards regulatory acceptance. The top arm here being the biological validation, understanding the biology behind the biomarker, and then demonstrating its application in case studies. And then at the bottom, we have the more technical validation, so analytical validation in the form of developing the SOPs, doing the ring trials, and demonstrating reproducibility. So we need both of these arms. I'm going to quickly give you a little bit, a really high-level background on the top arm, but we're going to spend a bit more time on the bottom arm. So many years ago, Georgetown with Heng Hong and Al Fornes and with Yuri Obrecht came up with this idea to identify this transcriptomic biomarker. And this was done in human TK6 cells. So this is a lymphoblastoid cell line. The group exposed the cells to 28 different prototype reference chemicals, half of which being DDI and the other half being non-DDI. And from that, machine learning was applied that identified 64 genes that are predictive of DDI potentials. And you can see the nice pattern up here with the agents on the x-axis and genes on the y, separating DDI from non-DDI. So here we have the TGx-DDI in vitro transcriptomic biomarker that predicts these probabilities. And we really like it. Over the last decade or so, we've been using it routinely. It has high specificity, and I'll show you that in a slide, to correctly identify these misleading or irrelevant chromosome damage assay results. It's based on an intact endogenous cellular response to DNA damage in human cells, so it's highly biologically relevant. It's transferable to other cell types, meaning we don't have to stick with TK6 because any cell line that expresses p53 signaling will be able to be used with TGx-DDI. And because we typically analyze TGx-DDI using whole genome sequencing, RNA sequencing, and similar platforms, it has multiplex capabilities with other transcriptomic biomarkers. And what I particularly like about it is that it's really hard to do pathway analyses. And having a biomarker like this is really objective. It removes the subjectivity of transcriptomic data analysis. So as I said, we've got the biomarker discovery, and then we move on to the context of use of the biomarker. This is the context of use that I'll be talking about the most today, that our working group is focused on, and that's its application in drug screening. So this is what, as you've heard about, we've been bringing forward to the FDA's biomarker qualification program. So in this context of use, TGx-DDI is used in weight of evidence analyses to inform irrelevant in vitro chromosome damage assay results. So we've got our suite of assays, we apply TGx-DDI, and we understand whether this positive chromosome damage assay in vitro is misleading or irrelevant positive. In my neck of the woods, we're more interested in chemical safety assessment, and there's all kinds of different ways that we can use TGx-DDI, and we are using TGx-DDI in chemical evaluations. So for hazard identification, for understanding mode of action, and for potency comparison, and I will show you a little bit about that. So that's the bottom context of use. But again, we're going to focus on the drug screening one today. Focusing in on the biological relevance. I've already mentioned this is biologically relevant to understand genotoxicity. Mechanistically, these 64 genes that I'm showing you here are enriched in genes under the regulation of P53, which controls the DNA damage response. So it's exactly what you'd expect, that the machine learning process has identified genes that make sense, that biologically plausible, that they should be good predictors for genotoxic outcomes. And as I mentioned, this requires an intact endogenous P53 response. And so we can apply to multiple cell types. We have been interacting with the FDA for many years now, and one of the first exercises that the FDA asked of the TGx-DDI biomarker team was further digging into the biological relevance of this. They wanted us to look at 42 different test agents that span different mechanisms of action and understand the outcomes of TGx-DDI in these test agents. So I'm just going to zoom in on 3 of the classes of chemicals that we looked at. This was originally all done with DNA microarrays. Now it's being done with nanostring and RNA sequencing, and other platforms like qPCR can also be used. So here we have our class 1 agents that are positive in the chromosome damage assay results and positive with Ames assay. And as expected, all of these compounds tested positive with TGx-DDI with this nice bright signal. Then we have class 4 here. These are our clean chemicals. They don't cause any genotoxicity. You can see that TGx-DDI, this is the overall call for TGx-DDI, is negative with 1 false positive. But what we were most excited about in this experiment was class 5. These are these misleading in vitro positives. And you can see that TGx-DDI effectively predicts these class 5 agents with high specificity. And then those case studies that I mentioned that were so important, we've done a lot of those now. For example, we've demonstrated how you can use TGx-DDI to show that caffeine is not a genotoxic agent. We've demonstrated how you can use it in combination with the micronucleus test and for potency comparison for chemical screening. And we've done a study where we demonstrate how you can de-risk agents with questionable genotoxicity findings shown here. And here in these studies, we're also demonstrating concordance across platforms and with other high throughput assays like the COMET assay. So I know what you're mostly interested about in this qualification, and so that's what I'm going to spend the rest of the time on. We have been interacting with the FDA since I think about 2012 maybe was the first face-to-face meeting we had with them. I might have gotten that wrong. But in 2016, you'll see at the top, the FDA invited our team to submit the full biomarker qualification package, and we did just that. And then we waited a year. But there was a little bit of a snag, a hitch here, and that was the introduction of the new 21st Century Cures Act that launched the new qualification process. But at that point, the FDA provided a letter of support saying, we're sorry, you have to start all over, but we really support you in moving forward with TGx-DDI. So let's just, you can see all these other things that went on, but let's just jump over to 2022. It took the following 5 years to agree upon the finalized qualification plan and our context of use that we were going to qualify, and then to launch the program. And while we were doing this, the FDA supported the project. Our fantastic HESI leadership team wrote a grant to the FDA, and we were successful in obtaining $250,000 of funding to conduct the ring trial. And so I'm going to tell you what that process has looked like for us now. So the goal of our qualification is to gain support from the FDA for a context of use of using TGx-DDI as part of a weight of evidence analysis. The goal is to retain the high sensitivity of the current test battery, but address the low specificity through TGx-DDI. And so you can read this here. It's a composite safety biomarker that provides additional mechanistic information to augment genotoxicity hazard assessment. It should be applied following a positive in vitro mammalian cell chromosomal aberration assay or an in vivo micronucleus assay indicating structural chromosome damage. All right, now onto the ring trial. The objectives that we determined with the FDA, what was missing was that bottom arm, and that's the cross-laboratory reproducibility of TGx-DDI classification calls. And so we decided to do this with one platform, NanoString, which was the preferred platform that Georgetown has been using extensively, Heng Hong Lee, and then using our resourcing from the FDA. And then at this point, we had willing volunteers who came on board, and this team has been working so hard, and I thank each and every one of them for all the effort they've put into this over the past about year and a half now. You can see all of them listed here and the different roles that they were playing. What we did was a study at 4 unique sites. Each of the sites received the test compounds that we were studying in solution. Each of the labs, and this is important, were assigned concentrations to use rather than doing range finding experiments. And obviously we're told which solvents to use. And these were all blinded. There were 14 chemicals. The work was done in TK6 cells, although we know TGx-DDI works in other cells. Everything was done in triplicate. And we produced, as part of the qualification plan, a really important part of that planning was producing the required standard operating protocols for the cell culture, the exposure, the analysis, and classification. So here's how it works. We've got TK6 cells. They're exposed to the chemical. TK6 cells don't express CYP enzymes. So we have to either expose with S9 if the compounds require metabolic activation. So they're either exposed for 4 hours without S9 or for 4 hours with S9, followed by replacing that media and a 3-hour recovery. A subset of cells are set aside for the MTT assay for cytotoxicity assessment, and that's done at 24 hours. Our top concentrations go up to 1 millimolar or solubility. But if they don't achieve that top level, the top concentration induce 50% cytotoxicity in the cells and culture. And this is consistent with the micronucleus assay guidelines. The 4-hour and the 7-hour time points are subjected to transcriptomic profiling using NanoString. And then bioinformatics software tools are used for the classification. I'll show you a little bit more about this on the next slide, but I want to highlight here, this is the TGx-DDI biomarker classification tool that was developed with the NIEHS with Jennifer Fostel. And this is a freely available tool that makes it so that you don't have to have any kind of understanding of coding in order to apply the TGx-DDI biomarker. So here's how the data analysis works. The labs all have a NanoString cartridge definition file and a reporter library file, and then a reporter count code or an RCC file. This is put into the software tool that's paired with the NanoString machines. It's called NSolver. And that software tool is used for the QA/QC using the built-in flags in that software. And we use that software tool for normalization and producing our extractable file that we then upload into the Manticore TGx-DDI software tool. That's the NIHS tool. At that point, there are 3 classification approaches that are applied. The first one is a near shrunken centroid probability analysis. And that's basically looking at those profiles of up and down regulated genes and saying what's the probability it matches DDI and what's the probability that it matches non-DDI. Then we have a principal component analysis that asks the new test chemicals, do they cluster with the reference training set, the reference set with the DDI agents or the non-DDI agents, and a similar analysis is applied to the 2D clustering. And so all the samples and concentrations are analyzed this way, and any one call that's DDI leads to an overall call of DDI for that compound. So after all of this time and all of this work by all these labs, this is the summary of the entire ring trial. It's I would have liked to have said it's completed now, but it's very close to completed. So these are the codes of the chemicals that all the labs received. Here's the decoded compound names here. This is the expected DDI call from each of them. These are the tests that met the cytotoxicity threshold. So let's just spend 1 second here on this column. You'll see there were 4 labs, but not all of the labs achieved the top concentration that we hoped they would achieve. So that means they fell short of the cytotoxicity limit, which was 50%. So that meant, unfortunately, that not all of these tests are actually valid tests. So we're a little bit short on that. You can see one of the compounds we didn't get up high enough for at any of the labs. So an important lesson here is, we had very limited time and resources to do this experiment, and that's why we pre-selected the concentrations. But moving forward, an extended concentration range or range finders is really what we need to do with TGx-DDI. But the fantastic news is, looking at these valid tests, here are the correct calls that were made using TGx-DDI for all the valid tests. And what you see at the top is 100% concordance with giving a DDI call, and a very high concordance with giving the right non-DDI call. We had 2 false positives for compound I and 2 false positives for compound O. And then our positive controls and negative control here worked as we expected. So overall, what does that mean? That's over here on the right. This is all our true positives and our true negatives here. And Andrew Williams did the statistics for us. We have a 100% sensitivity, an 84% specificity, and a 90% accuracy. And you can see the lower and upper confidence limits here. So quite happy to say the validation study, we think, has really been a success. First of all, we produced this SOP. It's quite a complex SOP, and all the labs were able to implement it and apply it and successfully conduct the TGx-DDI analysis. We had these pretty decent results. I'm very happy with them, especially because I would point out that the sensitivity and specificity of the in vitro flow cytometry micronucleus assay, which is really the go-to chromosome damage assay, are 90% and 65%. So we have an improvement on this assay using TGx-DDI. So I have a few more slides, but first, current status and next steps. We are really lucky, again, we have these generous team members who are willing to put in their time and resources to help us take this to the finish line. And we are grateful to Sanofi and Georgetown, who are donating reagents so that we can up the number of valid tests. So a few of those invalid tests are being reproduced now, and we're using actually range finding experiments to make sure that we nail that 50% cytotoxicity. So of course, then the next 2 steps will be to write this up in their qualification report for the FDA and prepare a manuscript on the ring trial. And this is where we're also excited to welcome a brand new team member. Our team, despite being around for a while, is still growing. So this is Dr. Vicky Wang, who has a PhD in pharmacology and environmental health sciences. And we're really happy that Vicky has come on board for the next 2 months working as a postdoc. And she's going to put 100% of her time over the next 2 months into writing these 2 documents up for us and completing the analyses in collaboration with the team. So welcome to Vicky. And then just to finish up, we love the TGx-DDI biomarker, but we have to bear in mind that there are other biomarkers out there, and the International Workshops on Genotoxicity Testing actually has a review that should be out really soon, summarizing all the in vitro gene tox biomarkers that exist. The 2 that emerge as the top performing players are TGx-DDI and GenoMark, which was developed in Europe, and work in this field is being led by Birgit Mertens at Sciences and her PhD student, who's now Dr. Tienpont, Anouk Tienpont. We collaborated with them because we know that TGx-DDI also works in HEPA-RG cells, and GenoMark was developed for HEPA-RG cells. We collaborated with them and used TempoSeq to see how concordant these 2 biomarkers are. Despite having different lists of genes, they're all under the regulation. Well, the majority of the genes are under the regulation of P53. And we had 100% concordance in calls for genotoxic or non-genotoxic between GenoMark and TGx-DDI. And this is actually the benchmark concentration for these 2 biomarkers in the confidence intervals. So you can see, even with a potency analysis in these cells, they completely overlap. So really great concordance between these assays. Also going on outside of HESI is work at Health Canada in using the TGx-DDI biomarker with Matt Meyer, Andrew Williams, Paul White. This is a cool paper from an amazing MSc student, Anne-Marie Fortin, who worked with me and Paul to develop an interpretation framework of where you might use TGx-DDI, for example, in a defined approach for determining mechanisms of action and genotoxic hazards. And the other really important thing that's happened more recently is the publication of an OECD test guideline for the GuardSkin transcriptomic biomarker. It's the first OECD test guideline for a transcriptomic biomarker. And there's a lot that we can learn from this. And when we look over what the GuardSkin guideline looks like, it's pretty clear that we have pretty much all of that because of our work with validating the TGx-DDI biomarker over the years. So much to learn from this process. So then to wrap up, we've extensively validated this biomarker. We've demonstrated that it works very well across different cell lines and transcriptomic biomarkers. And I know that, you know, every year you keep thinking that we're going to be done this, but I don't think we're quite done yet. We are in the final stages of biomarker qualification. But I think that this qualification and GuardSkin position us to go next to the OECD. The GenomeArk leaders have plans to go to the OECD, so we need to think about how to integrate with them, because I don't think we want to bring two separate transcriptomic biomarkers to the OECD. So I'm posing the question to the group now, is it time to think about bringing this project forward with GenomeArk? You may be tired, but I want to remind you that, as in the words of our famous Canadian basketball player, Marvin Phillips, the difference between try and triumph is just a little oomph. So that's my pitch to hopefully help encourage you to get involved. There's still time to help us take this to the finish line.
Really awesome presentation, as always, Carole. Thanks for doing this. Anne-Marie, did you have a question?
She may just be clapping.
Oh, okay. Maybe that was just clapping. Anyone else have any questions, thoughts? I agree with you, Carole. I think thinking of next steps in OECD, especially since we have so much of the data, is really important. Maybe a test guideline or some kind of IATA, but I think that's worth having the group talk about, but really open to hearing what others think about. I see Scott's hand is up.
Hey, Carole. Great talk.
Hey.
Great story.
Thank you.
Great story as always. One of the things I was wondering about, and the cross laboratory thing was something that caught my eye, and I hadn't really thought about it before. But batch issues, and how are you guys navigating the batch issues? I know that might be digging a little bit too deep for this, but I imagine it's a very realistic challenge when you're running on 4 different nanostring machines or something along those lines. So how are you dealing with that?
Everything is completely confounded. So the nanostring cartridges hold 12 samples. So all the controls for a chemical and all the concentrations for a chemical are on one cartridge. And so that's how we're managing that. So everything is compared against its cartridge matched solvent control.
Okay. All right. That makes sense.
Thanks, Scott. Time for one more question, if anyone has some.
I mean, I think that, as we bring this to the FDA, just linking back to Brett's proposal, I think having the reference library that we need for the proficiency testing that we'll be proposing for new labs when they adopt this technology will be important. So this is where I was, seeing that as a first obvious application.
Yeah, I totally agree. And I think too, some of the things we pointed to, and you had some of it in your talk, but having a CRO offering, having the tool available that is pretty user-friendly, and all our different labs were able to navigate and do, trying to make it as easy as possible for the adoption. And I think that applies to some of our other projects within eSTAR as well.
Yeah, and I think, the FDA, if that's where people want to go with this, I don't think that it's very different from the OECD, but at least there's a nice template now, and we sort of know what we're doing, so we can help other people moving forward with this.
Great. let's give Carole a big round of applause. Thank you so much for being a champion of this project and all your hard work and enthusiasm.